Today I'll present to you a process of DNA amplification used by many genetic technologies, polymerase chain reaction. To begin, a definition. The polymerase chain reaction, or PCR, is an almost entirely automated method of replicating DNA that allows researchers to target and amplify a very specific sequence within a DNA sample. The history of PCR. The PCR was developed by Kary Mullis in the early 1980s. The method was first formally presented at the American Society of Human Genetics Conference in October of 1985, and the first clinical application for PCR, an analysis of sickle cell anemia, was published the same year. So profound was the impact of PCR that Kary Mullis was awarded the 1993 Nobel Prize in Chemistry. PCR reagents. There are 5 basic reagents that are necessary for PCR: template DNA, PCR primers, nucleotides, tag polymerase, and PCR buffer. The template DNA is known as the starting DNA, which is being amplified during a PCR reaction. Because of the process, only a small amount of DNA is required for analysis. PCR primers are short fragments of single-stranded DNA, 15 to 30 nucleotides in length, that are complementary to DNA sequences that flank the target region of interest. The primers are typically used in pairs, and the DNA between the 2 primers is amplified during the PCR reaction. DNA molecules consist of monomer subunits called nucleotides. Specifically, deoxynucleotide triphosphates are the nucleotide bases added to the growing DNA strand by the DNA polymerase. This DNA polymerase is isolated from the bacterium Thermus aquaticus, which lives in hot environments and requires biomolecules that are heat stable. Therefore, tagged DNA polymerase can efficiently synthesize DNA under the hot intensive conditions of the PCR reaction. PCR buffer is necessary to resist change in pH and create optimal conditions for activity of tagged DNA polymerase. PCR buffers are often available in 10 times concentration and are sometimes tag formulation specific. The PCR process can be summarized into 3 steps. Note that researchers must first prepare 2 primer sequences. The primer nucleotides have a sequence that is complementary to the 3' end of each strand of the sample DNA molecule on either side of the DNA target sequence. First, the sample DNA fragment is placed in a solution along with nucleotides and primers. The solution is heated to break the hydrogen bonds between the base pairs, which causes the DNA double helix to open. This is routinely accomplished at 95 degrees Celsius for 30 seconds. The next step involves the PCR primers finding their complementary targets and attaching themselves to those sequences. Here the choice of temperature is largely determined by the melting temperature of the 2 primers. Again, the usual duration is 30 seconds. Finally, the last step is the polymerase extension step, during which the DNA polymerase is producing a complementary copy of the target DNA strand. The DNA polymerase starts adding nucleotides from the primer sequence in the 5' to 3' direction to the daughter DNA strands. In just over a minute, both DNA strands are replicated, resulting in 2 copies of the original target sequence. The usual temperature of this step is 72 degrees Celsius, considered to be a good optimal temperature for thermostable polymerases.
The polymerase chain reaction is a method for making many copies of a specific segment of DNA, starting with a very small amount. This technique can be used to identify specific microorganisms from a small amount of DNA and to identify persons involved in crimes from DNA on cigarettes or in a single hair follicle. The DNA to be amplified is mixed with deoxyribonucleotides, a thermal stable DNA polymerase called Taq polymerase, and DNA primers. The DNA primers hybridize to the ends of the gene to be amplified and provide a starting point for the Taq polymerase. The mixture is heated to break the hydrogen bonds in the DNA, forming single-stranded molecules. The mixture is then cooled sufficiently to allow the DNA primers to anneal to each end of the segment to be copied. Taq polymerase then synthesizes the complementary strand of DNA using the primer as the starting point. The temperature is raised again to separate the DNA strands and then lowered sufficiently to allow the primers to attach. Taq polymerase now synthesizes another set of new complementary strands. This process is repeated until enough DNA has been produced to be identified or used for further research. After 21 cycles, one molecule of DNA can be amplified to over 1 million copies. This amount of amplification can be achieved by running the reaction overnight in a thermal cycler, an instrument that automatically raises and lowers the temperature at appropriate time intervals.
There are many applications of polymerase chain reactions. PCRs are used by evolutionary biologists. PCR techniques can be used to sequence and analyze DNA that has been taken from long dead or even long extinct organisms. Then, the DNA can be analyzed and compared with DNA sequences of other organisms to help determine evolutionary relationships. PCR in the medical field. PCR can provide information on a patient's prognosis and predict response or resistance to therapy. Many cancers are characterized by small mutations in certain genes, and this is what PCR is employed to identify. PCR is extensively used in analyzing clinical specimens for the presence of infectious agents, including HIV, hepatitis, and malaria. PCR is also used in the analysis of mutations that occur in many genetic diseases, such as cystic fibrosis. Because of the sensitivity of PCR, this can be done from a single cell taken from an embryo before birth. PCR raises a number of ethical issues relating to amniocentesis, confidentiality, and personal integrity. Amniocentesis uses a hollow needle inserted into the uterus to scan for developmental abnormalities in the fetus. When the DNA is acquired from the embryo, it can be replicated and used to scan for potential birth defects or diseases. Because non-invasive samples may be used for DNA testing, such as a toothbrush or hair, people can be tested without their knowledge or consent. The question, what limitations should be placed on prenatal testing, arises when discussing many genetic technologies. The future of PCR. By tinkering with variables such as chemical reagents and pH, researchers have already reported success at copying larger pieces of DNA, including the entire genome of HIV. Extraordinary miniaturization of the hardware is also underway, as experimenters screw PCR onto chip-sized devices. In summary, researchers use PCRs to target and amplify a very specific sequence within a DNA sample. PCR can be performed using a variety of tissues and organisms, including blood, skin, hair, saliva, and microbes. Only trace amounts are needed to generate enough copies to be analyzed using conventional laboratory methods. That's the end of my presentation, and thanks for watching. I hope you learned something new about polymerase chain reactions and genetic technologies in general.